Good morning and welcome to Immunic's fourth quarter and year-end 2021 earnings call. My name is Jessica Brew, Head of Investor Relations and Communications at Immunic. I will also be the moderator on today's call. Speaking on today's call are Dr. Daniel Vitt, our Chief Executive Officer and President, as well as Glenn Whaley, our Vice President Finance and Principal Financial and Accounting Officer. For the Q&A session of today's call, we also have our Chief Medical Officer, Dr. Andreas Müller, on the call. Please note, all participants will be in listen-only mode, and this event is being recorded. After today's presentation, there will be an opportunity to ask questions. If you joined the webcast via the Zoom platform, there are two ways to submit questions. You can either submit your questions in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function of the Zoom portal to queue your question. If you joined today's webcast by phone, please press *9 to queue your question. Before we begin, I would like to remind you that this presentation may contain forward-looking statements. Such statements can be identified by words such as may, will, expect, anticipate, estimate, or words with a similar meaning, and such statements involve a number of risks and uncertainties that could cause Immunic's actual results to differ materially from those discussed here. Please note that these forward-looking statements reflect Immunic's opinions only as of the date of this presentation, and it undertakes no obligation to revise or publicly release the result of any revision to these forward-looking statements in light of new information or future events. Please refer to Immunic's SEC filings for a more detailed description of the risk factors that may affect Immunic's results and these forward-looking statements. I would now like to turn the call over to our CEO and President, Dr. Daniel Vitt, to begin the presentation. Daniel, please go ahead.
Yeah, thank you, Jessica. I would like to welcome everybody on Immunic's year-end 2021 earnings call. I do not want to start this call without expressing our sympathy with the people in Ukraine and expressing our solidarity with them. Despite of these developments, earlier this morning we announced our financial results for the year end of December 31st, 2021, and highlighted recent activities as well as upcoming milestones to our clinical development pipeline. During today's call, we will talk through our fourth quarter 2021 and subsequent highlights, financial and operating results, as well as anticipated milestones. As Jessica noted before we close the call, you will have the opportunity to ask questions. 2021 was another year of tremendous achievements for Immunic, marked by significant clinical progress across key pipeline programs, clearing the way for several important data readouts this year that are potentially transformative for the company. Notably, during the fourth quarter, we enrolled the first patient in our phase 3 ENSURE program of vidofludimus calcium in patients with relapsing multiple sclerosis. We also completed enrollment in our phase 2 CALOS-1 trial of vidofludimus calcium in patients with moderate to severe ulcerative colitis. We expect top-line data for the induction phase of the UC trial to be available in June of this year. For our second program, IMU-935, we reported positive unblinded safety, PK, and PD data from the healthy volunteer portion of our ongoing phase 1 trial. We also expanded the trial as planned to treat patients with moderate to severe psoriasis. Beyond this, we also initiated an open-label phase 1 dose escalation trial of IMU-935 in metastatic CRPC, and expect clinical safety data to be available in the third quarter of this year. Finally, we hope to see the first clinical data from the SAD/MAD part of the ongoing phase 1 trial of IMU-856 in the third quarter of this year, and expect to initiate the third portion of the trial in patients with intestinal barrier functions associated diseases during the first half of this year. That quick overview said, let me now walk through the fourth quarter 2021 and subsequent highlights in greater detail. Given the milestones we have achieved so far, our continued pace of development and ongoing interest in our key therapeutic focus areas, in October, we appointed Patrick Walsh to the newly created role of Chief Business Officer. This is a key position within Immunic, and Patrick has already proven to be a valuable addition to the team as we work to realize the full potential of our clinical programs. Also in October, we started treating patients with moderate to severe psoriasis in Part C of our ongoing phase 1 trial of IMU-935, representing the first time patients have been treated with our potentially best-in-class oral IL-17 inhibitor. To enable the rapid conduction of the trial, despite COVID-19 related limitations in Australia and New Zealand, where the trial is exclusively performed so far, we have early initiated measures to randomize patients faster. This includes a potential expansion of the trial to one or more countries in Europe. Based on our modified projections, we expect initial results from the psoriasis patient cohorts now to be available in the second half of 2022. Rounding out October's news, the key announcement was that we enrolled and randomized the last patient in our phase 2 CALDOS-1 trial of vidofludimus calcium in patients with moderate to severe ulcerative colitis. At completion of patient recruitment, the trial had randomized a total of 263 patients into 4 arms, 3 active dose arms of 10 milligram, 30 milligram, and 45 milligram, as well as placebo. We currently expect the top-line results of the induction phase to be available in June of this year. Backed by promising results from our previous phase 2A entrance trial, performed in UC and Crohn's disease patients, and the interim analysis of our CALDOS-1 trial, published in September 2019, along with vidofludimus calcium already established strong safety and tolerability profile, we believe that the drug could become a preferred oral treatment option for patients suffering from ulcerative colitis, an obvious alternative to biologics. In November, we enrolled the first patient in our phase 3 ENSURE trial of vidofludimus calcium in RMS. This was followed by enrollment of the first patient in the program's twin ENSURE 2 trial 2 months later in January. We have targeted an enrollment of approximately 1,050 patients in each trial. Dosing will be either 30 milligram daily of vidofludimus calcium or placebo. The primary endpoint for both trials is time to first relapse up to 72 weeks. Enrollment in these twin phase 3 studies comes on the heels of initiating our supportive phase 2 CALIPER trial in progressive multiple sclerosis. Together with these programs, mark a significant milestones for Immunic, in particular, as we have now initiated our first phase 3 program. As we have noted before, based on the strong activity observed in our phase 2 emphasis trial in RMS and the drug's well-established safety and tolerability profile to date, we believe that the design of the ENSURE program provides a straightforward path towards potential regulatory approval of vidofludimus calcium in RMS. Despite the limitations of currently approved therapies, the global MS market exceeds $23 billion, and vidofludimus calcium is uniquely positioned to address the unmet need for MS patients. Moving on, in December, based on the strength of preclinical data highlighting the therapeutic potential of IMU-935, to affect castration-resistant prostate cancer, we enrolled the first patient in an open-label phase 1 trial in this indication. The trial's principal investigator, Dr. Johann Sebastian de Bono, is one of the world's leading experts on the subject of CRPC. His expertise and deep understanding of the unique mechanism of action of IMU-935 provides important corroboration of this program within the scientific and clinical communities. Also in December, we reported positive unblinded safety, tolerability, and PK data from the single and multiple ascending dose portions of our phase 1 clinical trial of IMU-935 in healthy volunteers. The data showed a very attractive profile for IMU-935 and are consistent with our preclinical data supporting our vision of establishing IMU-935 as the potentially best-in-class oral IL-17 inhibitor. Additionally, we announced newly available preclinical in vivo data confirming that IMU-935 maintains normal thymocyte maturation in relevant acute and chronic mouse models. These data are consistent with previous preclinical in vitro data and support that we may have the first clinical stage Rho gamma T inverse agonist which circumvents thymocyte maturation issues. To our knowledge, such an outstanding selectivity hasn't been reported for any other Rho gamma T inhibitor so far. More recently, just earlier this month, we strengthened our IP position with the receipt of notice of allowance for composition of matter patents covering IMU-935 in the United States, in Europe, and in Australia. These patents provide patent protection into at least 2038, with further extension possible through potential PTE in the US and SPC in Europe, respectively. Also in February, we presented preclinical data on the potent anti-inflammatory activity of vidofludimus calcium at the 17th Congress of ECHO. Highlights included, first, that vidofludimus calcium reduces pro-inflammatory immune cell response by inducing regulatory macrophages, reducing pro-inflammatory cytokine secretion, and reducing T-cell proliferation. Second, that vidofludimus calcium shows an additive to synergistic effect with anti-TNF antibodies. And finally, that DHODH is important in the fraction of cells that receive a strong immune stimulus and are highly metabolically active. In conjunction with the ECHO Congress, we also announced the blinded baseline characteristics of our phase 2 CALDOS-1 trial of vidofludimus calcium in UC. Patients in the trial had active, moderate to severe disease, and we were happy to see that only 17% of the patients were pre-treated with biologics. The trial employed a central independent reader to evaluate the endoscopy eligibility criteria. At baseline, 55% of patients had a modified Mayo endoscopy score of 3, and 45% of patients had a score of 2. We believe that these data of randomized patients and the methodology regarding endoscopic assessment used in the trial contributes to ensuring an optimized study readout. In our 10K file this morning, we also released final data of cohort 2 from our phase 2 emphasis trial of vidofludimus calcium in RMS. We believe that this data set provides additional support for the previously determined dose selection for the ongoing ENSURE and CALIPER trials in RMS and PMS, respectively. Recall that our emphasis trial comprised 2 cohorts. Cohort 1 compared the efficacy and safety of 30 milligram or 45 milligram once daily vidofludimus calcium with placebo in RMS, while cohort 2 compared the efficacy and safety of 10 milligram once daily vidofludimus calcium with placebo in RMS. Full data from cohort 1 was published in the 3rd quarter of 2020, while 12-week interim data from cohort 2 was released in the 2nd quarter of 2021. In the newly available cohort 2 data set, the anti-inflammatory effects of vidofludimus calcium at the 10 milligram dose were observed to be lower than those found with the 30 milligram vidofludimus calcium dose in the pooled cohort 1 and 2 data, providing further support for the selection of 30 milligram dose in the ongoing ENSURE trials of RMS. The final cohort 2 data also provided evidence of dose proportional neuroprotective activity. For instance, the highest decrease of the biomarker serum neurofilament light chain was observed with 45 milligram dose of vidofludimus calcium versus placebo, with minus 26% median of difference between percentage change of serum neurofilament light chain. A substantial decrease was seen with a 30 milligram dose with minus 18%, while the smallest decrease was observed with 10 milligram dose of cohort 2 with minus 9%. The 10 milligram group of cohort 2 also showed a signal with respect to improvement in EDSS, consistent with those signals seen with the higher doses of cohort 1. However, all of these early signals need to be confirmed in larger patient populations with longer follow-up periods. Taken together, these observations suggest that higher doses, such as 45 milligram vidofludimus calcium, may be preferred doses for clinical trials in which neuroprotective effects are the main mechanism for improvement, such as in PMS. While the blinded treatment of cohort 1 was completed right before the COVID-19 pandemic started, final cohort 2 data provided additional evidence that ongoing vidofludimus calcium treatment may reduce the risk of COVID-19 infections. In the entire cohort 2 population of 59 patients, incidental COVID-19 infections in the active treatment group was 88.5%. Were less frequent than in the placebo group with 25%. Additionally, we recently obtained new preclinical data underlining that vidofludimus calcium shows potent anti-EBV activity at concentrations of 3.3 to 30 micromolar in a superinfection assay. A poster with the full data was presented at the ACTRIMS Congress in October. We also confirmed that vidofludimus calcium can be detected to a noteworthy degree in the CSF of animals after oral dosing. We believe that this finding suggests that vidofludimus calcium may be able to act directly within the central nervous system. For the next part of today's presentation, the financial overview, I would like now to hand over to Glenn.
Thank you, Daniel. We will now review the financial and operating results for the year ended December 31st, 2021. Let me start with the cash overview. We ended the year with 86.9 million in cash and cash equivalents, and also raised an additional 16.2 million through our at-the-market facilities so far in 2022. We anticipate this cash balance to be sufficient to fund operations through the first quarter of 2023. Regarding the operating results, research and development expenses for the year ended December 31st, 2021 were 61.1 million as compared to 38.6 million for the same period in 2020. The increase in costs for the full year reflect the continued ramp up of clinical expenses related to our 3 clinical programs, as well as increased personnel expenses related to the hiring of more people to support the company's growth. The increases were partially offset by decreased costs related to our phase 2 clinical trial in COVID-19 that was finished in the first quarter of 2021, and a decrease in drug supply costs for IMU 856. General and administrative expenses were 13.3 million for the year ended December 31st, 2021, as compared to 10.3 million for the same period last year. The increase in cost was primarily due to non-cash stock compensation expense, as well as smaller increases in costs across numerous categories. Net loss for the year ended December 31st, 2021 was approximately 92.9 million, or $3.93 per share, based on approximately 23.7 million weighted average common shares outstanding, compared to a net loss of approximately 44 million, or $2.81 per share, based on 15.7 million weighted average common shares outstanding for the same period in 2020. I would like to remind everybody that our 2021 net loss was impacted by the settlement agreement our subsidiary, Immunic AG, signed with 4SC AG in March 2021. Immunic AG settled its remaining obligation of a 4.4% royalty on net sales of vidofludimus calcium for 17.25 million. The payment was made 50% in cash, 50% in shares of Immunic stock. No further payment obligations remain between Immunic and 4SC AG. With that, I'll turn the call back over to Daniel for an outlook on our upcoming clinical milestones. Daniel?
Thank you, Glenn. As mentioned in the beginning of the call, we have a very exciting year 2022 with several significant milestones coming up, and I'm happy to walk you through these. One of our most important milestones in 2022 will be the readout of our phase 2 CALDOS-1 trial in patients with moderate to severe ulcerative colitis. We currently expect the top-line results of the induction phase to be available in June of this year. For our phase 1 clinical trial of IMU-935, we expect initial results from the third portion in patients with moderate to severe psoriasis to be available in the second half of this year. The initial data will provide us with the first important look at IMU-935 safety and efficacy profile in this patient population. In addition, we expect initial safety data from our phase 1 dose escalation trial of IMU-935 in progressive metastatic CRPC to be available in the third quarter of this year. As a reminder, the trial is designed to establish a recommended phase 2 dose and to assess safety, tolerability, anti-tumor activity, biomarkers, and pharmacokinetics of IMU-935 in this patient cohort. Finally, we anticipate unblinded safety data from the single and multiple ascending dose parts of the phase 1 clinical trial of IMU-856 in healthy volunteers in the third quarter of 2022. Initiation of the third portion of the trial in patients with intestinal barrier function associated diseases is expected in the first half of 2022. This brings us to the end of our formal presentation. Jessica, please open the call for the Q&A session.
Thank you, Daniel. We will now begin the question and answer session with Daniel, Glenn, and Andreas. As a reminder, if you joined the webcast via the Zoom platform, there are two ways to submit questions. You can either submit your questions in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function of the Zoom portal to queue a question. If you joined today's webcast by phone, please press *9 to queue a question. At this time, we will pause momentarily to assemble our roster. Our first question comes from Jessie Foe at Piper Sandler. Jessie, please unmute yourself and go ahead.
Hi, team. Thanks for taking our questions. This is Jessie on for Yaz. I had a few on CALDOS. Based on the baseline demographics on Friday, what do you guys expect to see placebo rates for clinical remission and endoscopy? And what aspects of the baseline do you expect to drive down placebo responses?
Hi, Jess, this is Andreas. I thank you very much for the question. I think we were very happy to provide you the data with the baseline characteristics because we believe we have worked hard to find a patient population where we have a very good chance of showing the properties of IMU-838 in the right setting in ulcerative colitis patients. What the baseline data showed that we had an active population. You can see this, for example, in the fecal calprotectin data, but also in the amount of patients or proportion of patients that had a very high stool frequency and had blood in stool to a very large degree, almost in every of the stool occurrences. So I think that, to my mind, the high activity seen in the baseline, I think, is the major driver for giving the drug a chance to show activity in this trial. On top of it, I think since the primary endpoint is a composite out of the endoscopic healing and symptomatic remission, we are also very happy that 60% or more than 60%, almost 60% of these patients had a Mayo endoscopy score of 3, which is also, I think, important to limit the placebo response in any IBD trial, because it is known that in IBD you have a placebo response, including sometimes for endoscopy, and having patients with a very extensive baseline disease in endoscopy, I think, allows us to limit the placebo response as well. So overall, I think we've been quite happy with the baseline data that we have presented, and we believe that this enables us to give our drug a chance to really shine in these in this study. And I hope, Jess, I addressed all of your questions and all of your aspects.
Yes, thank you, Andreas. I had one more follow-up. On the if there will be a phase 3, do you plan on running it yourself or with a partner?
I think that's too early to tell.
Okay.
We do, however, are in the middle of preparing, of course, how a phase 3 would look like. We're having a lot of very tight discussions with our medical advisors in IBD. We are very happy that we have a very large and also very well-known group of medical advisors for our IBD program. And over the years, we really had a very close relationship with them, and basically in anticipation for the study readout, of course, have a lot of ongoing discussions with them that target also, I think, the positioning of the drug, given what happened in IBD treatment over the last year or so, with the repositioning of the JAK inhibitors basically behind, more or less, as a medication, behind biologics as a medication of last resort. And also, I think we wanted to understand a little bit whether ozanimod, the newest entrance into the market, had more or less been observed to be in the same positioning as JAKs for IBD patients as well. And I think that also the discussion about the positioning of a drug that has a placebo-like or a very favorable safety profile in IBD patients as compared to these previous entries of other oral drugs. How does this change the perception and also positioning of the drug? I think that was also very important to us in terms of planning a phase 3 trial. So this is all ongoing, but I think it's too early to tell how we do it, because I think you need the data for that as well, and also with whom we're doing it, whether alone, with a partner. I think that's still open.
Okay, thank you so much, Andreas.
Thank you, Jess.
Thank you, Jesse. Our next question comes from Andreas Argyrides at Wedbush. Andreas, please unmute yourself and go ahead.
All right, good morning. Can you guys hear me?
Yes, good morning.
Oh, good morning. All right, thanks. 2 quick ones from us. So from the full emphasis trial results, could you provide some of your thoughts on and explanations on the lessened reduction in lesions, but more robust change in the neurofilament concentrations at 45 mg compared to 30 mg? And then just, I don't know if I missed it earlier, you mentioned a delay in Part C for the psoriasis trial. Just additional color that would be appreciated. Thank you.
All right. No, thank you, Andreas. I will address the first question maybe first, and then maybe let Daniel talk a little bit about Part C for IMU-935. So in the emphasis trial, which is, I think, for everybody to understand, is the phase 2 trial of IMU-838 in relapsing remitting MS, where we had reported fantastic results from the cohort 1 with 30 and 45 milligram. We added a cohort 2 that explored 10 milligram and placebo to really understand a little bit better the dose response curve. And we had published the interim analysis with week 12 data in early 2021, which confirmed, I think, the dose for relapsing MS that we are using in phase 3, 30 milligrams. So I think, first of all, the data in the cohort 2 readout now that we published this morning, is confirming, I think, our conclusions for the dosing. What was interesting, which I also find personally very interesting, is that we knew that from previous DH/ODH drugs in MS, that they have a particular strength on the long-term readouts, on the variables that are responsible for the long-term outcome of patients, specifically disability worsening and brain atrophy. So things that are related probably more in terms of the EDSS readout, how much disability you acquire over time in the long term for these MS patients. And the interesting thing that I find very interesting personally is that in the cohort 2 data with 10 milligram, you see a substantial effect in EDSS, and you see a substantial effect in, for example, disability worsenings. This is all, I think that the study is too small and doesn't have a long enough follow-up to really make conclusions. However, I think it gives us a first hint that I think the effect of IMU-838 on disability accumulation over time is potentially, again, I think very much in parallel to other DHODH drugs, is very strong. Whereas the anti-inflammatory effect that is more or less related to lesion reduction, I think is very strong at 30 milligrams, but we haven't seen a strong effect as much in the 10 milligram dose. So I think there's some, these results confirm that a drug like IMU-838 in terms of efficacy will likely have its strength in neuroprotective effects and effects that are measured in disability accumulation or potentially also in brain atrophy. And in that respect, I think these data for me are very exciting as compared to just having an anti-inflammatory effect. And I think that's very important, I think, for the positioning of the drug in the future as well. So I'm handing over to Daniel regarding the part C.
Maybe just one comment to what you said, Andreas. I think for us, it was really a positive surprise to see the EDSS data from the 10 milligram cohort. This was unexpected, honestly. And I think that speaks for the strength we have seen here relating to NFL data and so forth and all of that. So, but let's come to 935, to your second question. I think it's an obvious question. I think we follow our philosophy of being transparent, being early, and if you know us for some time, we usually try to react early if we see things not 100% aligned with our expectations, and therefore we decided quite early. This patient cohort of 935 trial just recently started, so but we felt if we really don't want to have substantial delays, we should add or could consider adding another country just to have a broader set of dermatologists contributing patients. So that's something we decided, and therefore want to share that with the market. I would not call it a delay.
Okay, all right, appreciate it, guys. Thank you. I'll step back in the queue.
Thank you, Andreas. Next question comes from Gobind Singh at JMP. Gobind, please unmute yourself and go ahead. Gobind?
Working?
Yes,
yes, now it's working.
Great. Sorry about that. Well, congrats on the pipeline progress. I guess my question would be, if I heard you guys correctly, I think there is the announcement that the interim results from the phase 3 MS trials and the phase 2 MS trial for progressive MS, those are new and happening this year. So I was just wondering if you're hearing anything from your clinical sites that might add some color around the enrollment speed, and can you remind us what the bar is in terms of whatever kind of clinical results you're going to be presenting? What would be results that you guys would be excited and continue development?
Yeah, hi Gobind. I'm not sure I totally understand the remark about the timing of the interim analysis for phase 2 and 3. I have not seen anything in our releases that would even talk about the interim analysis. So I'm not sure, maybe that was a misunderstanding with some of the things. So just to confirm, the phase 2 study in progressive MS will have its interim readout when half of the planned patients, we have 450 patients, so when 225 patients are enrolled, and these 225 patients have reached week 24 to have enough data, because we will basically report on neurofilament data as the interim readout. And then for the phase 3 trials, we said that we will have an interim analysis for the phase 3 in relapsing MS when half of the events, meaning relapses, have occurred in this study. In terms of the phase 3 interim readout, we have to look at the number of events and the frequency of events in this trial, and it's way too early to do this. So there's really no a more precise timing of the interim analysis that I can give you at the moment. We need a little bit more enrollment for that and a little bit more follow up on these patients. And but just to confirm, none of these interim analysis is expected in 22. Your other questions about, I think, enrollment, I think we are very happy to have seen a good enrollment now in all of our MS studies which are recruiting. We have 3 studies that are recruiting, the phase 2 study in progressive MS, and then Ensure 1 and 2, which are in relapsing MS, and they're recruiting very well, and we actually progressing with them as anticipated. So I think at this point, that's the only update I can give you.
Thank you for that. I think that was my bad on the milestone slide there. Just one follow-up for me as well on the MS studies. There's some recent studies that have actually been coming out about the role of EBV in MS. It's becoming more and more. It looks like that this causative relation, and you presented some data I think recently, and today you reminded the audience about your data in EBV with 838. Can you just help us connect the dots here in terms of how you think this is going to play out, and if you could relate that at all to the BTKs or any of the other, the CD20s or any of the other drugs that are popular commercially or in development, and if they have any role on EBV, that would be great.
Yeah,
and I'll hop back in queue.
Yeah, Goben, I think this is a very good question. And we have been, as a team, very excited about seeing more data and more information coming out about the role of EBV in MS. And there was always kind of the suspicion that EBV plays a role. And I think it became a lot more concrete now what the role of EBV is. So I think the study that made the most splash, I think, in public is that there was the study in several thousands of US military that were followed over, I think, 25 years or so, and looked at how many of those developed MS and what basically was known about their health status and before that. And the interesting thing was this study, for the first time, could make a clear link between an EBV infection that becomes neurotropic, meaning you see before the with the EBV infection, you see an increase of neurofilament at the same time. And those patients who have an increase in neurofilament at the time of EBV infection develop MS. And those, of course, I think 98% or so of the population has an EBV or made an EBV infection. But those who had an EBV infection and which was not neurotropic, but that basically did not show a neurofilament increase, basically did not develop EBV. So I think there's now a clear link between the EBV infection, having a neurotropism of that EBV infection, and developing MS. So that's the onset. It doesn't necessarily have to say that EBV plays a role during the disease. It's the onset. But then there was also very interesting data that came out, probably had made less of a splash, but I think were as equally important by a Stanford group, also in February, published in February of 22, that showed that they looked at the CSF, so cerebral spinal fluid, of patients during relapse. And it was always known that the CSF of patients in relapse contained some oligoclonal bands of immunoglobulin, but they did a lot more thorough analysis where this oligoclonal band of immunoglobulins comes from and what it is. So they found that, first of all, they have plasmablasts in the CSF at that time of relapse that are actually producing cross-reactive antibodies, and that's the oligoclonal bands. The cross-reactive means there's actually EBNA1 antibodies, but they're cross-reactive to a antigen on glia, microglia in the brain that's clearly linked to neuronal destruction. And so these cross-reactive antibodies are increased, produced by these plasmablasts. The interesting thing, you ask about different interventions that probably could influence, these plasmablasts. The interesting thing is that these plasmablasts were shown to be CD20 negative, which is kind of important because we're always thinking of EBV infections as these viruses are hiding in B cells. And you would think that, for example, anti-CD20 therapies or BTKs should be effective in eliminating EBV because they eliminate B cells. At least from this publication, that these plasmablasts are CD20 negative, they concluded that you have to find other therapeutic strategies to really have a EBV-free B cell recovery. So that's also, we believe it's very exciting for us because it could open the possibility of using, for example, IMU-838 with a established broad antiviral activity and also a EBV activity to help in terms of EBV-free recovery, for example, when you think about de-escalation after CD20 therapies and so forth. So I think this is all hypothetical at this point, but I think the hints that we get from this publication, I think are very exciting for a drug like IMU-838. This needs a little bit of time to spread, I think, the scientific community and probably a few more confirmations of this, but at least the data that came out about EBV were very exciting for Immunic and for vidofludimus calcium.
Thank you.
Thanks, Govind. Our next speaker is Matt Kaplan of Ladenburg Thalmann. Matt, please unmute yourself and go ahead.
Hi, good morning. Can you hear me?
Yes, good morning.
Good morning.
Great. Well, congrats on the progress. Just a quick follow-up to the last question. If we remain on that topic, how does the concentration that you studied and saw in vitro in terms of the 3.3 to 30 micromolar correlate with the concentration that you're seeing with 838 in vivo and in the CSF, in the central nervous system, since you're getting across the blood-brain barrier?
Hi, Matt. So I think, thank you for this question. I think we have seen a lot of splash by companies that do BTK inhibitors, claiming that they're the only drugs that really have a centrally acting a medication for MS. And we always suspected that statement is probably not entirely correct, as some of these smaller molecules should enter the CSF and the central component as well. So these are preliminary data. We're working on expanding this and having more data and confirming a little bit more closer to the human situation, what concentrations we'll get. To answer really fully your question, I think that you have, is that are these concentrations enough to make a meaningful contribution also in terms of efficacy from the central component? When you look at the relationship between serum concentrations and CSF concentrations, we see that we're at least on par, I think, with the data that were published with the BTK inhibitors. And we actually believe that the BTK inhibitors are not that unique in terms of being as a small molecule to act in the central component, central compartment, and it may not be, what sets them apart. So I think that, to me, was the main rationale for showing these rat data. But we will continue to generate data that will show that I think IMU-838 or vidofludimus has access to the central compartment as well.
Okay, very good. And then A question in terms of data that we should be looking for in the psoriasis cohort for 935 in the second half of this year. Can you give us a sense in terms of what you're looking for potentially in the efficacy profile of the drug and what you're modeling there and what we should be expecting?
Yeah, so I think we'll take in a little bit, we're learning a little bit from history. History in terms of the different drugs that had a full phase 2 program in psoriasis, where you look at how quickly, for example, things like the PASI score change over time. Usually the phase 2 trials are done with a 12-week endpoint or a 3 months endpoint. And you see that after 4 weeks or after 1 month, you have a half maximum effect in terms of PASI as compared to 3 months. So I think you have, the good news is I think you can very well estimate what the change in PASI score, not in the PASI 75 or so, but in the actual PASI score, what the change in actual PASI score, the average change should be. So when we look at this, I think you see that maybe the IL-17 antibodies or IL-23 antibodies, they're in the range of 30 to 35% change that they have at week 4. With Vitae Pharmaceuticals, the ROR gamma inhibitor some years ago, they found in their trial in moderate to severe psoriasis patients, I think they had 2 dose groups, and I think one dose group was somewhere around 20% change of PASI, and the higher dose was, I think, 28% change in PASI. So I think that's also the expectations that we have for our drug, that we want to show that we're in the same range, somewhere between the Vitae compound and the IL-17/23 antibodies. Of course, we measure much more in this trial. We do biomarkers. We have a skin biopsy where we do immunohistochemistry. We look at other clinical scores, like, for example, an itch component, which I think is very important also to see early efficacy. But the PASI score is usually the most well-recognized assessment in psoriasis, and everybody knows this. So that's why I think it's also, for us, I think it's the most visible outcome of this trial, probably.
And Matt, okay. One other comment to your first question, I think regarding the coverage, I think was also referring to the EBV activity.
Yes.
We were surprised that just by accident, basically, we have seen this benefit for preventing COVID-19 indication. That was a small cohort size and everything, but I think it was a quite remarkable, surprising difference between the active and placebo group on COVID, underlining the broad antiviral activity. And that also should hint to, for EBV, the concentration should be in the same range, and therefore we believe that that's covered. And the trough levels really cover that in vitro data, which we presented.
Okay, great. And just this last question in terms of, with the recent announcements and invasion of Ukraine, what's your exposure in terms of your clinical development programs in Ukraine?
So, of course, that has been some concern to us over the last weeks already. Yes, this is, I mean, it was an event that, of course, surprised the world. But I think we had looked at that. What we can say is that the CALDOS study, our study in ulcerative colitis, is completely unaffected. And the reason for that is that we had done, we have been very specific of doing the data cleaning for all of the data up to week 10, which is our primary endpoint, as quickly as possible. We have done all the monitoring visits. So that is all done. We basically have these data, and of course, it's not yet unblinded. So there are patients that are in extended induction, so we can't unblind the study yet. And that will go up to week 22. But all of the data that go into the primary endpoint are already cleaned, source data verified. So in CALDOS, we have actually no exposure. Ukraine had been part of other studies, of course, especially the MS studies are done in Ukraine as well. We have also started with countermeasures. You really, I think we have to look a little bit at this is a dynamic situation, and of course, this is very unfortunate, the situation, but we have to monitor this what kind of long-term repercussions this has for doing clinical trials in Ukraine. We have implemented some countermeasures ahead of time. We have to just see how this develops, and we anticipate that that's something that, given I mean, it's hard to expect at this early time how long this and how severe this conflict will be. But I think the best thing we can do at this time is monitor the situation day by day.
Okay, thank you.
Thanks, Matt. Next is Zekbe Jala of Roth Capital. Zekbe, please unmute yourself and go ahead.
Good morning, guys. Thanks for taking my questions, and clearly a nice cadence of data readouts ahead. So I know CalDOS is probably going to be of significant focus to investors. So just had a couple of questions. I think the first one is, I know the study enrolled biologically naive and biologically pretreated patients. So should we expect to see very different efficacy profiles in these patients? And then similarly, she would also expect to see differences in efficacy profiles between patients with different baseline Mayo scores coming into the study.
So this of course, I can only take the history because this can differ from drug to drug, occasionally based on the mechanism of action. Traditionally, I think the biologically experienced patient had shown less of response to active treatment than the biologically naive patient. So that's, I think, the general wisdom in IBD, let's say it this way. So that we include a lot of biologically naive patients, I think very consistent with the likely positioning of this drug, is to me is good news, because it gives the drug the chance to really show a good activity versus placebo, to my mind.
Thank you. And then just another follow-up here. What are your thoughts regarding the differences in the MOA of ozanimod versus IMU-838? And what would you expect to see in terms of time to response between the two different drugs in the induction phase?
So ozanimod as an S1P modulator has a very different, of course, mechanism of action, and you can see this on several things. For example, you see so S1P modulators basically more or less trap lymphocytes in lymph nodes or in other lymphatic tissue, and leads to a lowering of basically leads to lymphopenia in blood, and you can see the effect very clearly in all of the trials, and that's part of the mechanism of action, which leads, of course, to, for example, infections, virus reactivations, because you limit the ability of the immune system to respond. There's also now, just at ECHO last week, there were presentations, for example, how vaccination efficacy and humoral response against SARS-CoV-2 infection is much lowered for patients using ozanimod. All of this is not the case for IMU-838 because we have very different mechanism of action. We have a very selective effect only on what we call the bad guys of the immune system. Yeah, so the hyperactivated immune cells that are causing the autoimmune disease, and we leave the basically innate immune system and the immune response against infections completely unaffected. And we have seen this now in several trials where we've seen that the rate of infections, for example, versus placebo control is not increased. And some of the studies was actually slightly decreased. So I think that may be to the differences in MOAs. The phase 2 study and phase 3 study, I think, used an induction period of 8 weeks in ozanimod. I hope I'm correct. I've seen so many presentations last week at ECHO that I'm at an age where I can be confused sometimes, but I think it's 8 weeks. So we use 10 weeks. It's it's something that sometimes is doesn't make a lot of difference whether it's 8 or 10 weeks, because often these curves in clinical trials in terms of response also have this kind of exponential curve. Most of the effect happens in the early weeks or in the early 2 or 4 weeks. And the way you define induction period often is to give the chance for the drug to work in all of the patients, maybe patients that are responding slower. We know from our phase 2A trial, entrance trial in corticosteroid dependent patients, that the ability to withdraw corticosteroids was very quick. Within the first 2 weeks, I think you could lower, it to 20% of the baseline dose. So this is all published, but so we believe that also our mechanism of action leads to a very good and fast onset of the disease. And the time points 8 or 10 weeks is really doesn't make a lot of difference. It's just that you in a clinical trial you want to give the drug a chance to also work in most of the patients.
Thanks, Andreas. And I don't want to put words into your mouth. So just for clarification here, you do expect a fast onset of action. And then in terms of the magnitude of response, do you expect that to be similar, or should we be looking at it more as a composite, meaning the efficacy safety balance? Because you did mention how you expect 838 to come out better on the safety.
So I think maybe to start with the back, the last statement, I would totally agree that we always expect to be the safest drug in any indication.
Yes, clearly.
I think that we have learned now, and we're very happy about this. And that's basically the foundation on which we build our efficacy results.
And fast onset of action, I think that's also...
Yeah, fast onset of action, I think we have the data from the entrance trial that actually would indicate a very fast onset of this mechanism of action. What the expected efficacy results are, I think ozanimod, especially in phase 2, hasn't shown, overwhelming efficacy results if you look at other drugs. So you always look at this clinical remission, which endpoint that is very similar to or identical to what we have here in this trial. For some reason, we called it differently, but it was on a suggestion of the FDA. We call it a composite out of endoscopic healing and symptomatic remission, which is exactly the same definition as everybody's using for clinical remission. And you've seen that you have, data, let's say for tofacitinib, where you have only, 3 or 6%, depending on the dose, difference between active and placebo. And then you have the best drugs that probably have like 25% difference between active and placebo. And usually look at the differential between active and placebo. That's important. Ozanimod was more on the medium end. I think it was like 13% difference, if I remember well. But I think we would like to come out with a strong efficacy. And we have talked to our medical advisors, and I think very consistently they say for your safety profile, I think you have to have around 15% of difference between active and placebo to really have a outstanding efficacy.
Thank you. And then just a couple quick follow-ups here. So for folks that kind of like to change the goalposts, assuming that the data from the induction phase comes out really good, how do you think that de-risk the maintenance phase of the study? And then is it possible to make changes to the maintenance phase based on some things that you do observe in the induction phase of the study?
So the last question is very easy. No, the protocol is fixed. And also we do re-randomization for the maintenance phase, which is kind of usual in many IBD trials, because you expect that a maintenance dose may be lower than an induction dose, because here in the induction phase, you take sick patients very quickly from active disease to non-active disease, whereas in the maintenance phase, you maintain their non-active disease. And that has been done in several trials. Opaticitinib, for example, also had a very different dosing for the maintenance phase. So the re-randomization is important. In terms of going to phase 3, the induction data are really the important data, and this will decide whether you can go into phase 3. a phase 2 study, and that has been shown by many other trials, doesn't necessarily have to include the maintenance phase. And many phase 2 trials have not. We have decided to include the maintenance phase for the reason that we want that in phase 3, you have to show not only induction, but also maintenance of effect. So it is obligatory in phase 3, and we wanted to learn a little bit more about phase 3 planning, statistical effect size, and so forth. And that's why we included this in the phase 2 trial as well. But I would not, first of all, the readout that we have in June for the induction data does not include maintenance data because the maintenance is ongoing at that time. But also it will be available thereafter, but it's more or less a tool for phase 3 planning rather than a full-fledged efficacy readout for this study, for the phase 2 study.
Thanks, guys. And I'll save my final questions for a follow-up call, as I know we're approaching the hour here. Thank you again.
Thanks, Sekbe.
Thank you, Sekbe. So we have 2 more questions in the queue. Next one is Boobalan Pachapayapan at H.C. Wainwright. Boobalan, please unmute yourself and go ahead.
Good morning. Can you hear me okay?
Yes, good morning.
Hi, awesome. Congrats on your progress. So to start off the discussion, with respect to your CALDOS-1 trial design, I see that the 83% of the enrolled patients were biologically naive and 17% had prior biologic exposure. So I'm just curious whether the intention is to position IMU 935 after immunosuppressants, but before TNF-alpha inhibitors in the UC treatment paradigm.
So I think I would not draw that conclusion, but I think that would be a wonderful positioning, to be quite honest. In the discussion, I had said that we have a lot of ongoing discussions with our medical advisors. That includes, of course, discussion on positioning. And over the last 3, 4, 5 years, there have been several oral drugs that were approved or in late stage approval process for ulcerative colitis. And there was always the discussion where they should position. They aimed for the positioning to basically prior to biologics and after immunomodulators. And none of them really has achieved it. It had to do with the safety profile. And the FDA was very specific now, but all of the JAK inhibitors, and they actually named not only tofacitinib, they were very specific that this will apply the same positioning from their point of view. Regulatory positioning would apply to really only after at least you had one biological failure. So it's more or less also seen in the minds of the gastroenterologist as a medication of last resort. This will apply to all of the JAKs, including upadacitinib, that is specifically mentioned in that FDA communication as well. Ozanimod also has a considerable amount of safety challenges as well, and gastroenterologists see this as a disappointing entry into the UC market and the IBD market that will probably also end up with a more or less the same positioning as JAKs, which leaves kind of that therapeutic white space, yes, where everybody wanted to be completely open. The drug that is has a good safety, so you can use it early and use it in many patients early on in their disease, right after you used 5-ASA and corticosteroids, and before you use biologics. And I think that's when in our discussions with medical experts, they see as exciting for this program. The 83% biologically naive I think is a measure of maybe some of the countries and so forth that we were in. It was not intentional. I think we welcome it, but it was not intentional.
Got it. Thanks for the clarity. So there is a French company called Inventiva. It's also developing oral ROR gamma inverse agonist, and that's currently in phase 2b trial in psoriasis patients. So can you outline how your IMU 935 is differentiated from Inventiva's oral ROR gamma agonist, inverse agonist?
Thank you. That's a good question. And I'm not sure if this refers to Sidirogant, which is now in phase 2 by AbbVie, if you get it right. So this was originally developed by Inventiva, if I get it right. That molecule is, I think, an interesting compound now in phase 2. We have published a lot of data on our drug, tons of more data than ever published for the AbbVie Inventiva molecule, about mode of action, selectivity, exposure, PK, safety, and so forth. We're quite convinced that we have a best-in-class molecule, but let's wait a little bit until get more data for Sedirugand. And specifically would like to point to the publication we made in December. You can find that on the homepage. There was an update on specifically the selectivity of our drug, so that we selectively suppress IL-17 or Th17 polarization, differentiation. We suppress IL-17 in a very potent fashion in human lymphocytes, but totally absence of impacting thymocyte maturation. We have not seen any other company publishing such data, so we believe that this is a quite unique feature for our drug.
Great. And with respect to your ongoing metastatic CRPC trial, what do you need to see to move the program forward, and what are your go and no-go considerations?
So this is a phase 1 in CRPC, and we look at this as a phase 1 trial, where I think most importantly, we want to escalate dose, which is something that you want to do in oncology indications. And the dosing, of course, in oncology is expected to be quite different from immunology indications. And of course, you also want to have maybe a first hint of that things are happening. I wouldn't call it efficacy. I would call it more that you have a effect that you could translate into some sort of efficacy in future trials. And that's, I think, what we call expected phase 2 dose. It's a dose that is large enough, still be safe, but it's large enough to cause biologically effects that could translate into clinical activity in future trials. And you've seen this in some other phase 1 trials in oncology, that you're not looking for systematic efficacy yet, but you're looking for biologically effects that you can see at this dose that you can translate into efficacy in a phase 2 trial.
Okay, one final question from me. I know you spoke about interim analysis for your phase 3 ENSURE program. So I'm just curious, I know you'll be assessing event rates, but will you be looking at other endpoints as well during the interim analysis period?
No, we're not looking at endpoints actually in the interim analysis. We're looking at events rates and basically more or less the hazard ratio of events rates. Yes, whether our assumption what hazard ratio we get in this trial will be confirmed by this interim analysis. And really the idea is that you have to make assumptions here that drive the number of patients needed for this trial. I think we know from phase 2 already that, of course, you have an effect on lesions, you have an effect on relapse activity, even in the small population, but we need to see that our assumptions for the hazard ratio was the same. So we're not really assessing any specific endpoints in the interim analysis for the phase 3. We're looking at the hazard ratio and see how this translates into patient population or patient numbers needed.
That's it from me. Thanks for taking my questions and congrats on your progress.
Thanks.
Thank you, Boobalan. And finally, we have Ned Karenso, apologies if I pronounced this incorrectly, at SVB Leerink. Ned, please unmute yourself and go ahead.
Hi, this is Ned on for Tom Smith from SVB. We have 2 quick questions here. So the first one regarding the phase 3 ENSURE trial, what was the thinking behind the 18-month time point and the use of time to first relapse as the primary endpoint? And have a quick follow-up.
You said 18 months endpoint?
Yeah, 18 months time.
Okay,
72 weeks. Yep.
Yeah. Sorry. The idea of the 72-week follow-up period, I think, is looked at in terms of being a placebo-controlled trial, getting enough. Having enough chance for enough relapses to happen in a longer follow-up period, but having the follow-up period short enough to be able to do a placebo-controlled trial in as many countries as possible. So that's the short answer. And then time to first relapses. Time to first relapse. So the regulators ask for a relapse-related endpoint in a phase 3 study. And that's been traditionally also done in all the other trials. The guidances actually give you a choice of annualized relapse rate, ARR, which was traditionally used in phase 3 trials, or time to first relapse. Time to first relapse has become more interesting as relapse rates in MS patients are very well documented to go down over the last 2 decades and considerably going down, so that ARR assessment becomes more and more difficult to do with a reasonable number of study population. So I think there have been actually published reports that compare these 2 endpoints and basically really highlight the advantages of time to first relapse as an endpoint for relapse.
Got it. And another question regarding the phase 2 CALDOS-1 trial. Could you remind us quickly again, like what are the specific efficacy thresholds and clinical improvement to view this as a positive data set?
So I think you mean in terms of statistical planning? Just wanted to understand our statistical assumptions, more or less, how we look at the sample.
Like the efficacy threshold or the bar for success here.
Yes. So we have made statistical assumptions that are very much in line with what I just outlined as the expected delta. We also made assumptions, of course, we had to make some assumptions for the placebo response. Yes, so we know that all of for the clinical remission, and there's good summaries now available, and you look at other trials, the placebo response is basically 10% or less, often considerably less. And we have basically been conservative and basically assumed also a placebo response of 10%, and then basically added the delta. And we also assumed, for example, a dropout rate of 20%, and then we came up with basically the 240 patient sample size that we needed. So we've actually recruited 263. We had very good success, especially at the end of the trial, recruiting very fast. So we had a little bit over recruitment. And also I think we haven't seen the dropout rate. We had seen less dropout. So we feel very comfortable that I think the statistical assumptions, I think, will lead us to success in this trial.
And if it comes to effect size, maybe a comment from my end. If you look on the whole, the plenty molecules which are used there, and also on the biologics, if you have something like the effect size of vedolizumab or something like that, this would be a success, of course. And I think the biggest challenge in the last couple of years was more, as Andreas elaborated, on the safety concerns for drugs like JAK inhibitors, which were unexpected, and I think limiting the use there, specifically in this big gap between the current baseline medications like mesalazines and steroids and so forth, and the antibodies.
Got it. Thanks again for answering the question.
Thank you, Ned. And thanks to everyone for your great questions. This concludes our question and answer session. I would like to turn the conference back over to Daniel for any closing remarks.
Sure. Thank you, Jessica, and thanks to today's attendees for your insightful questions. We are very excited about the progress we achieved last year and the value inflection points we anticipate this year, including the phase 2 ulcerative colitis data for vidofludimus calcium in June, and the initial patient data for IMU-935 in the second half. With this, I would like to close today's call. Thank you very much for joining, and we're happy to answer any additional questions one-on-one.
Also from my side, thank you for joining Immunic's fourth quarter and year-end 2021 earnings call today. The conference has now concluded. You may now disconnect.